Thank you. I'd like to thank President Knapp for inviting me to participate in this forum. I found it really quite interesting. Usually I attend scientific meetings, but this is a little bit different, and I think I'm learning a lot more, actually. I've been asked to consolidate two lectures that usually take me about an hour each, and that's the relationship of academic medical research and industry, how these programs can collaborate and interact together, and to push this all down into 15 minutes. So that's really an important, difficult task, but I'll do my darndest. I'm a clinical pharmacologist. I have trained both in medicine and basic science. I've had positions all over the country, Virginia, Stanford, Texas, Northwestern. But I've also, while I've spent most of my career in academic medicine with primary appointments in the Department of Medicine and joint appointments in the basic science departments, I spent about 10 years in industry as a corporate officer and vice president of pharmaceutical research and development. And as you heard from Steve Lerman, I've helped colleagues co-found several biotech companies. What I want to do is to tell you about what's happening today in the excitement of biotechnology, academic research, and how they all need each other to make this technology move forward. The biotech industry was born shortly before I went to Stanford in the late 1970s, when Stan Cohen and Herb Boyer, on a napkin in a restaurant one night, decided that they could take genes and express them in bacteria for recombinant technology. That was the birth of Genentech. And of course, the biotech industry just flourished as a result of that for the past 25 or 30 years. Today, we can express all sorts of genes in all sorts of systems, not only bacteria, but in mammalian cells and even plants, and has a great deal of potential. The research interest that I've fallen in love with for my entire career has been cell communication, cell signaling. How do cells talk to each other? How does the brain control your kidneys or your gastrointestinal tract or your heart rate? These organs release molecules that we call messengers. And the first slide is going to show you a typical list of such messengers. It's not a complete list by any means, but these are molecules that today we call hormones, autocoids, paracrine substances, growth factors, cytokines. And the pharmaceutical industry has taken advantage of these molecules from this research information that came largely from academic programs to create products. And it's just a partial list. And what companies have done is not only take the native compound, but modified its structure to prolong its half-life, its distribution in the body, and even made analogs that block their activity for diseases where you're making too much of these molecules, such as some of the cancers or endocrine disorders. So this is an example of sort of what I do. I take these molecules and I figure out how they regulate the biochemistry and biology of their target tissue to cause a biological effect. And we've looked at lots of biological effects. We've looked at smooth muscle relaxation, regulation of blood vessels, airway smooth muscle, GI smooth muscle, secretion and excretion in the intestinal tract. We're now looking at stem cells and how they differentiate. We're looking at cancer cells and how we can prevent them from growing. And they're very interesting to sort out all this biochemistry. So while I'm a clinical pharmacologist and have done clinical research, I really prefer to look at the basic biochemistry of these pathways because I think it's a gold mine of information. And by understanding in some detail how these molecules communicate with tissues, we can take the pathways and dissect out molecular targets and discover new novel pathways and new novel drugs. And that's what I do. So I do clinical research, but mostly basic research. And often it's always related to some medical disease, with the understanding that at some point it could result in an interesting and important medical product. The industry to develop drugs in the past often took molecules off the shelf, administered them to an animal to look at a biological effect, whether it was lowering blood pressure, decreasing heart rate, some biologic effect. And if the animal had an effect and didn't die or didn't have a toxic event, this was a promising molecule that perhaps could be developed into a drug. And the pharmaceutical companies would take these compounds and make analogs so they had longer half-lives, they could be formulated, administered in various ways, intravenous or oral or whatever. And it was a very slow, tedious, and very costly process to discover drugs. When Boyer and Stan Kohn came along with their recombinant technology, this opened the doors now for an entirely different approach for drug discovery, which we call rational drug design. Now we look at the detailed biochemistry in tissues, and by identifying the pathways of communication in the biochemical pathways, we can take those enzymes and proteins and pull them out and screen them against libraries, huge libraries of compounds. Sometimes these libraries are thousands or even hundreds of thousands of compounds. I know one company that screened 1 million compounds against one of our targets that we were interested in to find a drug that's now in clinical trials for pulmonary hypertension in premature babies. So it can be done. It's a lot faster. However, once you get that lead compound, you have to come back and modify the chemistry to make sure that the half-life is adequate, that it's not going to be toxic. It's more potent. And the rule of thumb is that the more potent a molecule is, the less likely it will be toxic if it's very selective for a molecular target. But it takes very organized groups of people with lots of different disciplines to pull this off. Not only do you need biologists and pharmacologists and physiologists and cell biologists, but you need chemists, you need toxicologists to put this whole story together to create a product. It's very time-consuming, it's very expensive, but it's a lot faster, in my opinion, than doing it the old-fashioned way by putting chemicals in animals that took forever, often 10, 15, 20 years to create a product. Now we can find important potential products within a couple of years, but we don't know if they're going to be useful and applicable in human studies once we get to that point. But before I get into this, I want to point out a couple of concepts that some of you are aware of, but some of you are not. If you have a product that's going to be $1 billion a year sales, for every month you lose time in getting that product on the market, you've lost $80 million. $80 million pays for an awful lot of research in medical research. There are many drugs that are billion-dollar markets. There are some drugs that are $10, $15 billion markets, like the statins. So time saved is very important. You want to get there as fast as you can, and to do all the appropriate research to get that product out there that is effective and safe. Another point is that it's impossible to see toxicity in your preclinical studies, in your animal studies, or even in your first phase 1 or phase 2 studies in the clinic with these compounds if the incidence of toxicity is less than 1%. If it's 0.1% of the patients that get toxic, injures the liver, injures the kidneys, whatever, that means you have to give it to 1,000 patients to see one event. So sometimes you'll never detect toxicity until it's on the market and been given to a few 100,000 patients. And when you get to the market at that point and you run into a really nasty toxic effect and you have to take it off the market, it's cost you hundreds of millions of dollars. So you don't want that to happen. So what you want are molecules that are highly selective, hopefully with low toxicity, and very effective and potent on the targets that you're aiming them for. Toxicity is an impossible thing to predict. It's probably the most difficult aspect of pharmaceutical development of drugs. You can make them, discover them, make them more potent, do lots of tricks with them, But to predict that they're going to be toxic or not is almost impossible until you do those studies in enough animals or enough patients. I often tell technology companies being developed by some of my colleagues and friends that low-tech is faster, cheaper, and probably more profitable. High-tech is very high risk, but it's a biggie if you win. I mean, you can hit a huge market with big margins, but it's a very high risk. It's been estimated that less than 1 to 10% of the products that go into the clinic are likely to finish the clinic and be approved for marketing, a small percentage. I think the odds can be better than that. If you're really clever with a good research team, you can get there faster with more success rates. My success rates at Abbott were really quite high because I really think about research very seriously. I feel very comfortable with clinical medicine as well as basic research. So our success rates, I think out of 17 products in the clinic, only one really failed. All the rest did quite well. When engineers build bridges and skyscrapers, they do all of their homework. They think about the project from start to finish. The same has to be done in medical research. Is the target that you're going to aim this at some medical disease? Is it an important one? Is it a disease that requires some novel therapy where the current therapy is not sufficient or totally doesn't exist? Do you have all the resources to pull it off? Do you have the staff? Do you have the equipment? Is the intellectual property safe? Intellectual property is not just a single patent, but it's a fence of patents that protect all that technology and all the analogs and all the nuances of that approach, so that other companies can't invade your technology and come along and tweak the molecule and make it a little bit different and enter your market. Who are the competitors? How far along are they? Often very difficult to predict because industry doesn't publicize its research. It doesn't tell you what it's doing because it will provide clues to the competitors. So the only time you see companies publishing data, this is often the case in biotechnology companies because they're looking for funds and promotion to attract interest, but in the big pharma companies, they only publish when they've killed the project or they're about to market it and they want the publicity to influence the medical doctors to use the product. So you often don't know what they're doing. Is the product going to go for an orphan disease? An orphan disease is when there are less than 200,000 patients requiring that product. Some cancers, some genetic disorders. If it's an orphan disease drug, you can fast track it through the FDA for your clinical studies and you get more rapid approval to get it to the market. What's the realistic market projection? I've learned from my experience in industry that marketeers are very good at predicting markets once they have gone into the same market again with a related compound. They're very precise about what they think it'll sell, how many patients will take it, et cetera. If it's a novel approach in a novel disease where they don't have experience, they're not very good at it, frankly. And I've had many arguments with the marketing staff and senior management in companies saying, you're wrong. And I feel that I know more about the potential market size and profit margins than they do. The other question is, how far can you take it? Do you have the funds and resources to really take it all the way through to introduce it to the market? With startups, that's unlikely. It's been publicized that the big pharma companies can spend hundreds of millions of dollars. I've seen some numbers, $1 billion to develop a product. I don't believe that. I think the numbers realistically are in the $200 to $300 million range. I can tell you that in biotech companies, you can get products on the market after $30 million, $50 million, $100 million investments. So it is doable. But you've got to know that you've got to raise that money at the time you need it to move to the next milestone in your clinical development program. And you don't want to raise all that money up front because you dilute yourselves. So you raise it as you go, And you hope the investors are there the next time you need to go back to the trough and raise another 10 or $20 million. Now let me turn to my research with nitric oxide that Steve Lerman briefly referred to. Some years ago, I was interested in how hormones and drugs regulated smooth muscle motility, contraction, relaxation. And we accidentally discovered that an important drug, nitroglycerin, which was known to cause smooth muscle relaxation and lower blood pressure, it had been used for more than 100 years to treat angina pectoris, but not knowing how precisely it worked. We discovered that it was a prodrug or precursor that was converted to nitric oxide in the body. That was a novel concept. A free radical, a gas, was now going to be a messenger molecule to mediate the effects of an important drug. And we subsequently learned that not only is it mediating the effects of drugs, that I called nitrovasodilators, but it was also a natural product in the body that mediated the effects of lots of other hormones and messengers in all sorts of tissues. We published these results in 1977. That's the reason I went to Stockholm. But it turns out today, 35 years later, there are 130,000 publications in the field of nitric oxide research. And a very popular area in medical research, leading to lots and lots of novel drugs. And I'll review this for you briefly. We know that in blood vessels, this is a cartoon of a blood vessel, with the endothelial lining on the left-hand side of the slide, smooth muscle compartment on the right-hand side of the slide. If you take all of your blood vessels in your body and put them back to back, they'll go around the earth 2.5 times. It's a huge organ. The endothelium is a single cell thick, like an inner tube in your tire. And this is talking to the underlying smooth muscle to regulate the function of the smooth muscle. Here in red at the top in the middle is nitroglycerin, which forms nitric oxide to regulate the production of another messenger molecule, cyclic GMP. And I'm not going to go through the biochemical cascade. I don't want to get into too much detail with you and lose you. But the ultimate effect is that it relaxes the smooth muscle by influencing the modification of the actin-myosin filaments that slide together back and forth. By dephosphorylating myosin, the filaments slide apart and relax. When it phosphorylates myosin, they come together and cause contraction. There are drugs such as acetylcholine, a neurotransmitter, bradykinin, histamine, that will also cause relaxation, but only if the endothelium is intact. And that's because the receptors that recognize these molecules are only found on the endothelium and not on the smooth muscle. And when that happens, the endothelium makes nitric oxide as a messenger that moves across to the smooth muscle to cause relaxation. There's a disorder called endothelial dysfunction. Patients with hypertension, diabetes, atherosclerosis, tobacco use, and probably obesity have blood vessels that do not make enough nitric oxide. So consequently, the blood vessels in those patients are constricted, restricting oxygen delivery and nutrients to the tissues downstream. We know now that because of this endothelial dysfunction, we can now come up with some new approaches to treating these diseases to supplement the therapy with antihypertensives or insulin treatment in diabetes, et cetera. This is a partial list out of these 130,000 publications that are out there to show you how this whole field of biochemistry and biology can be utilized now to develop a lot of drugs for lots of other diseases. We know that nitric oxide is a neurotransmitter in the brain. It plays a role in the gastrointestinal tract. It regulates the heart. It regulates the kidneys. It participates in inflammation in the joints. It probably plays a role in Parkinson's disease and Alzheimer's. It regulates genes. It can regulate stem cells and to differentiate into various cell types. So it's got lots of promise now for rational drug design and development. And there are compounds being developed that will mimic these pathways or block these pathways. One of the compounds you are all familiar with, it's become a sort of a street drug, is Viagra. The nerves in the blood vessels of the penis release nitric oxide as a neurotransmitter to cause relaxation of the blood vessels to fill with blood. Viagra enhances the effect of nitric oxide. That's how it works in erectile dysfunction. There are other applications in pulmonary hypertension, in premature babies. Their blood vessels in their lungs are very constricted. If you let them inhale a little nitric oxide, they dilate those blood vessels. They stop shunting blood right to left. The hypoxic blood in the systemic circulation is improved. They're no longer blue babies. So there are lots of other potential applications of this whole field. And that's what I do. I really look for new ways to treat important problems. We now have some interesting data with cancer. We think we can treat some important cancers by manipulating the nitric oxide cyclic GMP pathway. Thank you very much for your attention.